go. Recording begins. This is October 4th, October 24th, Biology 105. Thank you all for making it. Don't forget, there's an exam on Monday. Bring a pencil and a Scantron 882E form. There are places to get the 882E form, such as the Rose House and the Testing Center. And who knows another one? Cultural Justice Equity Center. Yep, they have all got Scantrons for you, for free. All right? Very cool. Today, we are going to wrap up our PowerPoint on cell communication. And any time that we've got available after we finish discussing this, I'll just open up for study guide questions, Q&A for the exam, which, again, is on Monday. Very cool. Okay, folks, so we have been talking about signal reception. We've been talking about, mainly we've been talking about signal reception and then the different types of membrane receptors. We talked about intracellular receptors, which bind ligands that are small and hydrophobic. We talked about extracellular receptors or cell membrane receptors, right, which detect extracellular signals. Those signaling molecules are usually larger and hydrophilic, so they don't freely cross the phospholipid bilayer. Once the signal has been received, the signal must be transduced, all right? Meaning that the signal changes forms. This is what we talked about on Wednesday, last time, okay? We talked about a little bit of how signals can change forms. And when I say that, I don't mean that the signaling molecule necessarily gets morphed and changed in its shape. I mean that while the initial signaling molecule was this red balloon, all right, the next form of the signal is the high concentration of ions, or all of the phosphorylated proteins, all right? So, I mean, it changes forms in that it goes from being one thing to a different thing, but the signaling molecule does not actually get affected. Does that make sense to folks? Awesome. Okay, let's talk signal transduction. So the signal transduction pathway is not always so simple, where relay molecule 1 is activated, and then it activates relay molecule 2, which then activates relay molecule 3. There's complication here, where maybe the signal transduction pathway branches a bunch, where relay molecule 1 activates relay molecule 2A through 2F, and it elicits a ton of different responses, all right? Also, if you think about these activated relay molecules, they don't necessarily just activate one other molecule and then stop activating molecules. What I mean here is relay molecule 1 gets activated, and then for a period of time, it will remain activated, and while active, it's going to keep activating relay molecule 2. So that's going to naturally amplify the signal, right? You go from a single signal molecule to maybe 10 activated relay molecule 1s, to then each one of those activating 10 of relay molecule 2, and each of those activating 10 of relay molecule 3. So you get a bunch of activated molecules. So signal transduction, why would you even have it, okay? Why wouldn't the receptor just directly elicit the response? Well, because these signal transduction pathways allow for amplification of a signal, all right? And each step of the signal transduction pathway can itself be regulated. So if you don't have the relay proteins, for example, in one of these pathways, then that specific cellular response can't be elicited by that receptor. Or if you think about these as enzymes, you could regulate each one of those enzymes to buffer the response. So signal transduction pathways are complicated. They allow for two things in our purposes, really three. The pathways can branch, the pathways can amplify signals, and the pathways can be regulated. So it's another way to regulate the response of the cell to a given signaling molecule. I've got examples coming up for you So I hope they help. The biggest, the thing that we're going to focus on the most as far as signal transduction pathways here is going to be phosphorylation cascades. And phosphorylation cascades are a way where a signal gets amplified. So you go from the presence of a single signaling molecule and you end up with a ton of activated, usually enzymes. So you get a massive cell response from a very small signal. These phosphorylation cascades, very important. It does require that we know about the kinase enzymes, which I already talked to you about on Wednesday, and the phosphatase enzymes, okay? So kinase enzymes transfer phosphates from ATP to something. A protein kinase transfers phosphates from ATP to a protein. A phosphatase removes a phosphate group from something. A protein phosphatase removes phosphate groups from a protein. So think about them as working like, bless you, on-off switches. So if a kinase gives me a protein and phosphorylates me, then I become active and I do stuff. If a phosphatase takes that phosphate group away from me, then I become inactive and I don't do anything. On-off switch. Cool? Now think about this. Let's say that's the receptor. It's a receptor tyrosine kinase. All right? It receives its signaling molecules, it dimerizes, its kinase activity begins, and now it activates me by giving me a phosphate group. Now I have become activated. The kinase did its thing, and I've been phosphorylated. Now imagine that I'm also a kinase. All right? I don't have to give my phosphate group to this next molecule to activate it, because that's not what a kinase does. As a kinase, I transfer phosphates from ATP to protein, which means that while I'm phosphorylated and activated, I can go and put phosphates onto a bunch of other things and activate a bunch of other molecules. All right? And I'm just going all over the place, transferring phosphates from ATP onto molecules and activating them. That's amplification, okay? Because I went from one signal to a single activated molecule, me, and then I go around and I'm phosphorylating everything. I'm phosphorylating everything. All right? See how that's amplifying it? A question, please. A single activated protein phosphatase could deactivate many active molecules.
Okay, so they can do more than one.
Yes, because they're all enzymes. Okay. And remember, that's a key thing about enzymes is they're not used up when they carry out their reaction. Okay. That's a good way to think about it. Okay. Yeah. No problem at all. There's my laser pointer. All right, phosphorylation cascades. Check it out. Receive a signal. Activate a relay molecule, okay? This relay molecule then activates a kinase. This kinase then activates a kinase. This kinase then activates a kinase. This kinase then phosphorylates a protein, making it active, and that gives you a cellular response. Now, it's a phosphorylation cascade because you've got phosphorylation, 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 phosphorylation. And then each one of these enzymes, it doesn't just do its job one time. It's an enzyme. Remember, it's not consumed in the reaction. So it's this first active protein kinase. Maybe it activates 10 of these protein kinase 2s. Maybe it activates 50. Maybe it activates 100. All right? That's amplifying the signal in a phosphorylation cascade. All right? So say that this inactive protein is the actual enzyme that's going to break down, we'll use the example of breaking down glycogen. How does insulin work? Well, insulin works by activating enzymes that break down glycogen to release sugar into your bloodstream. Your body receives the insulin signal, and that activates the enzyme, the enzyme that breaks down glycogen. So if this is the glycogen enzyme, you could get a ton of these molecules activated, all right, from just a single molecule of insulin, because the signal gets amplified, so the response is larger. I've got an actual nice figure to illustrate that coming right up. So here's the phosphorylation cascade, where a kinase gets phosphorylated, then it phosphorylates a kinase, and it phosphorylates a kinase. Are we cool with the phosphorylation cascade? It's kinases activating kinases via phosphorylation. Yeah, I talked about that. Cool? Awesome. Stop it. Cool. Same thing. Now it's zoomed into each step. So if I'd talked a little slower, I would have been like, and thought this through a little more, I'd be like, let's look at it now on a step-by-step basis. Here it is. But I think I explained it enough in the single figure. Are we cool with this? This seems more complicated. This is the level I want you to understand a phosphorylation cascade on. Okay, cool. Get out of here. Go, 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 go. All right, cool. Second messengers. What is a second messenger? All right. Well, think about that ligand, that signaling molecule. Think about insulin. All right. That's the hormone that gets released in your body. That's the first messenger. That's the thing that's transmitting the signal of, hey, our blood sugar's low. Do something about that. That's our first messenger, okay? The primary signal. Now, second messengers, okay, second messengers are things that get produced inside of the cell. They get generated inside of the cell. They're water soluble, so they won't really move out of the cell, but then they diffuse throughout the inside of the cell, and they initiate another, other changes leading to the cell response. All right? The biggest one we're going to talk about is cyclic AMP. I'll show you an example of that in just a second. But the idea is, all right, I'm a receptor tyrosine kinase. I'm an RTK, or I'm a G protein coupled receptor. And what I do is I receive the signal on the outside of the cell. I'm a transmembrane protein, so this part of me is embedded in the membrane. And then I cause the production of something else inside of the cell. That molecule's stuck inside the cell, and it's going to diffuse and interact with other things, eliciting a response. So those are the second messengers, the things that get produced. You may notice, if you've been checking the dang old study guide, that I don't say the word second messengers on the study guide. I don't know. I'll do with that information what you will. Cyclic AMP is the most discussed second messenger, okay? Because remember, you've got ATP, you've got ADP, all floating around inside your cell all the time. So one of the problems with making second messengers is you need the starting molecule to produce the second messenger. But you've got ATP all over the dang place. So take ATP, remove two of those phosphate groups, make AMP, and then you kind of make it cyclic because there's two bonds to the phosphate group. And now this molecule can be detected by the cell, all right, and it can be used to elicit a response. The cell's like, oh, cyclic AMP levels are high. Let's start to break down more ATP, or let's ramp up the expression of this gene. There's too much variety to assign camp a single function. All right, and then there's another enzyme for converting cyclic AMP back into AMP, where then it can be built back up into ATP. All right, cool. So here it is in the flow chart. Put it in the flow here. You've got a G protein coupled receptor. The G protein coupled receptor receives the signaling molecule, its ligand. The receptor binds its ligand. This is its first messenger. It then puts GTP onto a G protein, okay, activating it. The active G protein goes on to activate the enzyme adenylyl cyclase. The enzyme adenylyl cyclase takes ATP, which is floating all over the place, all right, and converts it into cyclic AMP. Cyclic AMP is now the second messenger, and it can go and activate a bunch of other responses. All right? So it's something that got made inside the cell in response to the presence of the extracellular signaling molecule. I am not going to test you on anything having to do with cyclic AMP. I am not going to test you on anything having to do with second messengers. I will answer questions about it, but the sooner I finish this PowerPoint, the more Q&A time we have for exam questions, which is on Monday. Are there any questions? Yes?
I just want to clarify. So if we have an extracellular, like if you have a signal that cannot cross membrane and needs a ligand.
That is the ligand.
Okay, it's a ligand. But if we have something that can't cross the membrane, it's the first messenger.
Yes.
And then whatever it produces after that is the second messenger.
Yeah, essentially. It's intracellular because the signal needs to go into the cell to get the cell to do anything. See, that's the outside of the cell at the top. This is the inside of the cell here.
Yeah, no, the second messenger is intracellular, but the first messenger is not intracellular.
Exactly, 100% correct.
So it then becomes intracellular via second messenger.
The second messenger is like the intracellular signal.
Okay.
Yeah.
So that's how
Signal transduction.
and everything else we've been learning about that can't cross the membrane work.
Basically.
Okay.
Yeah, they make some second messenger. Now, it's not always this complicated. Sometimes there's just a kinase enzyme right here that gets activated, and the initial receptor starts off a phosphorylation cascade. Maybe there's not an actual second messenger. Maybe it's in the middle of the pathway. No matter what, it's going to be more complicated than what we talk about here.
Okay. So
It always is.
the receptor we're also talking about that changes shape after it's been signaled from the outside, that shape-changing receptor is a secondary messenger. Or
You're talking about the receptor tyrosine kinase, I think?
the one that it was on the slide, it changes shape. It has like the, because I know kinase has the phosphorylation.
Just stop me here.
Okay, if it's not important for the exam, then I'll stop.
Yeah, talk to me about it in office hours. Yeah, cool.
Okay, cool.
All right. No problem. Absolutely. I always want to answer more questions, but I also want to have some time for study guide questions. All right, cool. Good. Let's keep on crushing it. Next example of second messengers, okay? Calcium ions. It's an intracellular signal. So you've got an extracellular signal, okay? And normally inside of a cell, you've got, check it out, right? Calcium exists as an ion. It won't cross phospholipid bilayers. You've got this big endomembrane system made up of phospholipid bilayers. The cell is usually just actively pumping these ions into, say, the endoplasmic reticulum, or into the center of mitochondria, or into the central vacuole if it's a plant. Basically, the cell is usually sequestering ions in specific membranes, in membrane sacs inside of the cell, okay? The cell can regulate the concentration because the ion cannot freely cross the membranes. But here's the thing. If there's a huge flux of calcium ions into the cytosol, the cell can detect it and respond. So a ligand-gated ion channel right here, okay, that could be regulated from a receptor outside, it goes like bing, sends a signal right here, boom, that opens up, and calcium ions flood the system because they're going from areas of high to low concentration. You can get a really rapid response. This is involved in a lot of defensive pathways, so check this out. You've got the G protein coupled receptor. The G protein coupled receptor activates a G protein by attaching GTP to it. Activates an enzyme. This one's called phospholipase. What that does is it breaks down a phospholipid. All right? It makes this molecule that's a derivative of the phospholipid that serves as a second messenger. That molecule diffuses through the cell, hits a ligand-gated ion channel that detects it. That ligand-gated ion channel opens, and then calcium ions flood out. These calcium ions are also serving as second messengers because they are not the initial form of the cell signal. And then this influx or this increase in concentration of calcium ions is detected by the cell, which activates a ton of proteins, which elicits a huge cell response. All right? It's going to be really important if you talk, did I already say this? If you talk about like plant defenses to herbivory, calcium ion fluxes are one of the main defenses. Anyway, it's really cool. It's not on your exam. We can talk about it more on Wednesday, or come chat with me about it. It's cool stuff, I promise. All right, next, final thing, the response. We've talked about the reception of the signal, the transduction, and as I mentioned, the thing I want you to focus on is the phosphorylation cascade. That's going to come up again. And then the cell response. How does the cell actually respond? Oftentimes, it's through expressing a gene that wasn't being expressed before, or it's by regulating a metabolic pathway, activating or inhibiting an enzyme, right? Those are the two main cell responses that I'm focusing on here. Gene expression, change in cellular metabolism, cell growth or cell death. The last two cell responses are pretty self-explanatory, yes.
I got a question. I get why apoptosis occurs in multicellular organisms, but in single cellular organisms, why would it occur? Or why would a cell want to kill itself?
Yeah, absolutely, because natural selection acts on populations, not individuals. So if there's a population of single-celled organisms and it becomes adaptive for some of them to die at a certain point, let's say it becomes adaptive for if some of them have the genes for dying at a certain age, all right, if that makes the population survive better, it doesn't matter that that individual died because those genetics are encoded in the entire population. So you can have something that's bad for the individual, but is good for the population. And if that population succeeds more than other populations, then those genes get amplified.
Oh, so it's just it's beneficial to the population.
100%. That's exactly it. And that's one of the main arguments or theories behind why aging even happens. Because we don't have to age. There are plenty of organisms that don't die of natural causes, die of old age. Question.
I actually saw it was a video on the world's longest organisms,
Mm-hmm.
the world's organisms with the longest lifespan.
Yeah.
And I guess they had a hypothesis that aging was done because if you had a few immortal organisms, they wouldn't be evolving and they would be more susceptible to mass extinction.
100%. Yep. If you've got populations that are refreshing and their genetics are being refreshed, then it's more likely that they're going to be undergoing natural selection more. You're going to have them be more adapted to their environments. Yep. So there's reasons that aging and cell death, which is on the macro scale, could be like organism death, but there's reasons that death can be beneficial for the population, and that's what natural selection acts upon, is the entire gene pool. And that's how evolution occurs. It's not by the individual scale. Yeah. It can be bad for the individual, but good for the population. So sometimes you get a signal outside of a cell that tells the cell to grow, tells the cell to divide. Sometimes there's a signal that occurs outside the cell and follows one of these signal transduction pathways, and the cell's response is to die. All right? We're going to talk about gene expression changes in cellular metabolism. I've already talked to you about them a little bit. Let's start with the gene expression one. All right? So this is going to bring back up transcription factors. We talked about the process of transcription. Transcription is when DNA gets converted into RNA. And for our purposes, it's usually mRNA that we're talking about, but I'll talk to you about other types of RNA later in the course when we get into our genetics piece. So check this out. There's a receptor. A growth factor gets detected by the receptor. This growth factor is going to tell the cell, turn on genes involved in growing. All right? So the receptor activates a relay molecule, which triggers a phosphorylation cascade. Eventually, activating a transcription factor. Look, it was a kinase because it activated this by attaching a phosphate group. All right? Now that the transcription factor is active, the response from the cell is start making mRNA, start expressing the gene. So in this case, the response by the cell is gene expression. Also, cell metabolism. I've got a better image of this, I think, in our signal amplification example, but I might just be getting ahead of myself because I've seen these figures before. So check this out, right? You've got, in this case, we're starting with the second messenger. The second messenger camp is here. It's detected by these kinases, and it activates them, okay? It turns on these kinases, which then phosphorylate another kinase, activating it. Phosphorylates another kinase, activates it, all right? Then this enzyme right here is actually responsible for breaking down glycogen and producing glucose. Now, this is a little bit annoying because this is too accurate, you know? It's like too accurate. It makes it feel more complicated without clarifying a lot of stuff. So the point here is that you've got this enzyme that's now involved in breaking down glucose. The fact that it's a kinase is annoying, where it's like adding on a kinase group or a phosphate group. But the point here is that you've activated an enzyme now that's actually breaking something down that wasn't being broken down before. So the signaling molecule, okay, led to breakdown. All right, it led to the ramping up of a metabolic reaction or a change in cell metabolism. Is that okay? Awesome, love it. Cell growth, all right. There are a lot of these things called growth factors. You dump them on a cell, it makes the cell grow. We'll talk more about growth factors when we talk about regulation of the cell cycle, but they basically just make the cell get larger, accrue more cytoplasm, generate more organelles, try to divide, and then keep expanding. Growth factors make cells grow and usually lead to their division. Apoptosis, programmed cell death. All right? When you get signals that trigger apoptosis, which the receptors that detect them are called death receptors, which is pretty cool as far as the name goes, I guess. Maybe cool is not the right word, but yeah, you destroy the cell, it blebs off in this specific way, where it prevents the cell from just breaking open and dumping all of its cytoplasm into the surrounding area, which, as you could imagine, could be harmful. So it safely destroys the cell in this programmed process of apoptosis, which occurs during embryogenesis in order to develop digits. All right? So this is also a possible cell response. So just to reiterate, we had 4 different cell responses, and those were change gene expression, change metabolism, cause a cell to grow, cause a cell to die. All right? And all of these could be mediated by any of the different classes of receptors. There could be second messengers involved. It could be an intracellular or an extracellular receptor. All right, it's all this dang stuff, all this variability. Okay, signal transduction. What's one of the benefits? We can fine-tune the response. All right, so some things to consider about the signal transduction pathway. Check this out. We're going to focus on signal amplification a lot. Okay. So, epinephrine binds to a G protein coupled receptor. That's one molecule of epinephrine. All right. Epinephrine binds to a G protein coupled receptor that activates G proteins. It actually activates 100 G proteins. 10 to the 2 G proteins, okay? Now there's 100 G proteins activated doing their thing. Each one of them goes and activates a single adenylyl cyclase enzyme. So now we've got 100 active adenylyl cyclase enzymes. But then each one of those adenylyl cyclase enzymes activate or creates or fosters the production, catalyzes the production of 100 cyclic AMP molecules. So now you've gone from one single G protein, right, to what? 1,000, 10,000, 10,000 molecules of cyclic AMP. Each cyclic AMP molecule goes and activates a single protein kinase, so we have the same number. Each one of those activates 10 other kinases. Each one of those activates 10 other kinases, activates 10 other enzymes, glycogen phosphorylase. And what these enzymes do is take glycogen and release glucose, but they are also enzymes, so each enzyme can carry out this reaction 100 times. So you went from a single molecule of epinephrine to 10 to the 8 molecules of freed up glucose. All right? So that's signal amplification, and it's possible because these enzymes don't get consumed in their reactions, these kinase enzymes. So phosphorylation cascades are very important in signal amplification. That good? Awesome. Awesome. That's almost the last point on the study guide. There's one thing left on the study guide, and that's how different cells respond to the same signal differently. And that all comes from, well, like which receptors are present, okay? So in order for a cell to respond to a signaling molecule, that signaling molecule never enters the cytoplasm, right? So if a cell's going to detect a signaling molecule, it has to have the receptor for that signaling molecule. Only this cell's able to respond to a green circle, all right? These three cells can't even detect green circle. So if you don't have the receptor for green circle, then you don't respond to the green circle signal. Fair? Fair enough? All right. Next, there can be different types of receptors that bind the same ligand. So check out this cell on the left and this cell on the right. This cell on the left has a receptor that can detect red triangle. This cell on the right also has a receptor that can detect red triangle. But they operate differently in how they transmit the signal. So the cell on the left can have a different response from the cell on the right. It's a different receptor, but for the same ligand. So you can either not have the receptor, and if you don't have the death receptors, the cell won't undergo apoptosis even if the signaling molecule for apoptosis is there. All right? Or maybe there's a mutation in the death receptors, and when the apoptosis signal happens, it actually acts more like a growth factor, and now the cell's supposed to die, but instead it divides and grows. Okay? So the receptors, which are proteins, determine which signals the cell can detect and how the cells respond to those signals. All right? And remember, folks, all the protein instructions are encoded in the DNA. So one major determining factor, like what makes a different cell, okay, is what genes got expressed, okay, and what proteins are present in the membrane. Because that determines what signals the cell can respond to and how it responds to those signals. All right? Scaffolding proteins, they're not on the exam, it's boring. You got a bunch of these different like relay proteins, put them together. Put them close to one another, scaffold them. That makes it go faster. Duh, it's boring. All right? Different kinds of cells express different proteins. These different proteins allow cells to detect and respond to different signals. The same signal can have different effects in cells with different proteins and different pathways. Sometimes pathways branch or crosstalk, and this further helps the cell to coordinate incoming signals. You know I don't read slides, but this is a slide that's meant to summarize how important the different receptors are in cell responses. This is meant to summarize the information that you need for the exam question that has to do with this. Okay? So I just made a summary slide of text. Sometimes these pathways branch. Sometimes different receptors, you have to get 2 molecules, 2 ligands to get an actual response. All right, sometimes these branches crosstalk, these pathways crosstalk. There's all sorts of variability in eliciting the correct response from the cell to the presence of signaling molecules. Of course it's complicated. It's always complicated. Okay, and that concludes the week 9 PowerPoint. In week 9, we're not gonna start week 10. Instead, I'm just going to open it up to questions for the exam. If somebody's got one, just raise your hand. Yes?
So I just wanted to ask this, but are we going to have a practice exam from like an SI?
Oh, thanks for asking. No, doesn't look like. Oh, wait, no. There won't be a study session, a practice exam session, but Finn said that they'd be sending out a practice exam that they're writing. So you should expect.
It's like right in my eye.
This? It's unpleasant?
No,
This
very present.
this one. Okay, yeah, no problem. Yeah, no worries. Let's talk about like this. Can I actually just, this could be really fun. Document camera. I'm falling. Whoa. Yeah, that's really bright and unpleasant. Is this helpful? So everybody, focus. All right. Let's try you now. Okay, too much fun. No more fun allowed. The answer to your question is there will be a practice exam. It should show up in your announcements as a linked file. I'm sorry for blinding those of you who are in this portion of the classroom. Next question. Yes?
It's sort of all multiple choice.
Yeah. It's still all this multiple choice. 25 multiple choice questions, 3 points each. Study guide's posted. Yeah. Yes?
And what's on the study guide is what the exam focuses on. So if it's not on the study guide,
If it's not on the study guide, then you don't have to worry about it for the exam. You know, you should still learn it because it's all so fascinating, but not for the exam. Yes?
So the last question on the study guide is phosphorylation cascades and enrolled protein kinases, which I know we just went over.
Totally.
But protein kinases go through phosphorylation cascades. Do all phosphorylation cascades involve protein kinases?
Yes.
So protein kinases and phosphorylation cascades are the same thing.
Whoa. They are intrinsically linked. The kinase is the enzyme that's actually going to like phosphorylate, and then phosphorylate, and then phosphorylate. And the phosphorylation cascade is I activate, this kinase is inactivated, I activate it, okay? And then it goes and it activates this kinase. It stays activated, that's important. And it's activating kinases. And then these kinases go and activate additional kinases. And that's the phosphorylation cascade.
So phosphorylation cascades have to do with kinases.
Yes, the kinase is the player.
Okay, so they're intertwined.
Intertwined.
Okay, great.
Solid.
Yes.
Oh, yep, we got one over here, yes.
it's not cumulative, right?
No, it is not. Only the things on the study guide. So in any of my classes you take, I always make exhaustive study guides. And if you can ever show me on an exam, like, John, here's the study guide, here's the exam, this question was not on the study guide, I will not only give you credit for that question, I will give the entire class credit for that question. So confident am I in my study guides being exhaustive. Additional question. I thought I saw one there and then there. Yes.
Yeah, I was just wondering about the sink sources and purpose of oxygen.
That's a big one.
Yeah.
Good call. So it's sinks and sources of oxygen and photosynthesis and respiration, right?
Metabolism.
Oh yeah, cool. Metabolism and photosynthesis. Cool. Perfect. So let's go ahead and take a quick look here at this similar figure. Okay, we're going to keep it a little bit simple by using these simplified figures. In cellular respiration, oxygen gets consumed at the end of the electron transport chain, where it serves as the terminal electron acceptor. It's able to serve as the terminal electron acceptor because it's so electronegative, right? So there's oxygen as the terminal electron acceptor. In respiration, it's being consumed. So it's a sink, right? Now in photosynthesis produces oxygen. So it's a source of oxygen, right? And that oxygen gets released in the light reactions when water gets oxidized. So if you remember, in the first step of photosynthesis, the first step of the light reactions, let me get a good diagram for you. Photosystem 2 gets excited by light, and that kicks an electron off P680, generating P680 plus, which then oxidizes water to release oxygen. But the study guide just says, what's the source and the sink? The source of oxygen in photosynthesis is from water. It's from the oxidation of water. The sink of oxygen in respiration or metabolism is as the terminal electron acceptor in the electron transport chain, where it generates water. Of course. And you had a question next. Yes.
Someone that also likes to review past lecture slides. What's the earliest week of slides that will be on the test?
Fantastic question.
Well, this week is also going to be on.
This week is also on the test. So this exam covers membranes, energetics and enzymes, respiration, photosynthesis, and cell communication. The earliest one being, I believe, membranes. Yep. So you start with the membranes because there's, I think, one question that relates to the characteristics of an integral membrane protein, which has to do with it being hydrophobic and also having at least one region of hydrophilicity so that it could actually be embedded in the membrane hydrophobic domain. I got one here and then I'll get to you next. Yes, deal? Yeah.
Will we need to know, like, off the top of our heads, oxidizing and reducing agents for the exam?
Yes. Well, you need to have the whole picture, otherwise it'll fall apart in memorizing. So remember, like, a reducing agent reduces something. An oxidizing agent oxidizes something. And meaning that a reducing agent puts electrons onto something, and an oxidizing agent takes electrons off of something. If you have that piece, it's going to be important because you need to know that, like, NADH and NADPH are reducing agents, but that NAD plus and NADP plus are oxidizing agents because they take electrons. Cool?
Yes.
Awesome. Awesome. Yes, please. Where respiration happens? Yeah. So cellular respiration occurs within the mitochondria, and it occurs, well, the first step, glycolysis, occurs outside of the mitochondria. Let me be more specific. Respiration, boom, right here. Okay? So in the cytosol, we've got glycolysis, right? Where glucose gets broken down into pyruvate, and then pyruvate actually enters into the mitochondria. So then in the mitochondrial matrix, you've got pyruvate oxidation and the citric acid cycle. That completes the breakdown of glucose, releasing carbon dioxide. You should also know that, like, where's CO2 come from? Okay, comes from citric acid cycle, or the Krebs cycle. After this, the majority of the energy is all held in these reducing agents. I'll use the terminology since I told you it's important. In the reduced form of NADH and FADH2, most of the energy is held here. That energy then goes into oxidative phosphorylation, where those electrons on NADH and FADH2 fall to a low energy state in the electron transport chain, eventually hitting oxygen as the terminal electron acceptor, and the energy lost by these electrons is used to pump protons into the intermembrane space. So they get pumped out of the mitochondrial matrix into the intermembrane space, and the electron transport chain, so both stages of oxidative phosphorylation, occur in the inner mitochondrial membrane. You pump the protons, you generate the proton motive force, and then in the last step, I promise. Chemiosmosis here. In the last step, ATP synthase harvests the proton gradient. So the most direct source of energy for synthesis of ATP is the proton motive force, or the proton gradient across the inner mitochondrial membrane. Cool? And that's in respiration. Remember, cellular respiration is all about just making ATP. Awesome, yes?
What's the purpose of fermentation?
Fermentation regenerates NAD+. So fermentation exists so that you can keep desperately running glycolysis. All right? So glycolysis takes glucose and breaks it into pyruvate, and that gets you a little bit of ATP via substrate level phosphorylation, right? So you get a little bit of ATP. If there's no oxygen present, then you can't run your electron transport chain. The whole thing just backs up, all right? There's no place to put these electrons, so the whole electron transport chain backs up if there's no oxygen, and so you can't do chemiosmosis. So you desperately try to get some energy from glycolysis. But in order to run glycolysis, you need an oxidizing agent. You need something to pull the electrons off of glucose. And that oxidizing agent is NAD+. But all your NAD+ is NADH, because it had no place to put its electrons because you got no oxygen. So your electron transport chain is entirely backed up. So all you have is NADH. And if all you have is NADH, you can't do glycolysis. So fermentation, this step right here, this process right here, you take the NADH, you put the electrons onto pyruvate, and that gets you NAD+. And then that NAD+ is used to oxidize glucose, get you a little bit of energy. Solid? Okay, awesome. Next, we got like 2 minutes. Yes?
What does it mean by osmosis based on percent ion content?
Totally. So I've given you osmosis questions. They're in the B4, but I usually talk about concentrations as like molar concentrations. So this example right here of mammalian blood has the equivalent of 0.15 molar sodium chloride. Seawater has the equivalent of 0.45 molar sodium chloride. But if these were percentages of sodium chloride, it's exactly the same principle, right? Water would still flow towards the area that was 45% sodium chloride from the area that was 15% sodium chloride. I just didn't feel right having a question on the exam that relates to percent solutions without mentioning it. So it's just like this, but instead of molarity, it's percent concentrations. Water flows towards high areas. I saw your hand first. Go for it.
So just to go off that question, that question is going to be based on like isotonic, hypotonic.
Yep.
type stuff.
Water flows toward the high number. You can just memorize that and you'll answer that question right. We got time for one last question. Do you still got it locked and loaded?
I was just gonna double check that what I wrote was right, and then you answered my question.
That's how freaking good I am. I answer questions before they're asked.
Oh, that's how good I am.
Oh, okay. I love it. That's very good. But that part's not on the recording, so nobody knows that part. Great work. Great work to all of you. You made it to a Friday Bio 105 lecture in week 9. Like, that's incredible. That's really great work. That's the end of today's lecture. See you on Monday for the exam. Best of luck. Use the study guide, folks. I'm going to end the recording here.